talk about our efforts in the energy harvesting, especially using some of the new materials that are of interest in further improving the efficiency and cost. So let me give a brief introduction into this area of microbial or bioelectrochemical systems. So we have seen a surge in a lot of interest and reports in recent years in how microbes can generate power. There have been numerous articles, such as the one back in 2023, microbes. Just some excerpts here. In the long run, real potential is replacing bio-responding drugs. Electric microbes, one of the 50 best inventions of 2009, selected by Time magazine. And then it has been selected, this microbial fuel cell contest is selected as one of the top 10 new water technologies to save the world. Now, what precisely is bioelectrochemical systems? So it's based on an electrochemical system in which biocatalysts, specifically microbes, or in some cases enzymes also, perform oxidation and reduction at electrodes. So you can think about a system that has microbes that are electrochemically active, that takes wastewater out of organic materials as an input, and has in the production side maybe electricity production, biohydrogen production, or chemical production, at the same time capable of wastewater treatment and bioremediation, as well as some interest in utilizing this concept as an environmental sensor. Now, within that, there are many different types of bioelectrochemical systems, and the two systems I would like to focus today is microbial fuel cell and microbial electrolysis cells. So microbial fuel cell is a type of a bioelectrochemical system that produces net electric power utilizing various organic substrates through direct electron transfer. So there's really no fermentation process involved. The way it works is a typical microbial fuel cell has two chambers, an anode chamber and cathode chamber, having an electron each side, separated by a protein exchange membrane. In the anode chamber is where the electrochemically active microbes are living. So they can utilize the natural resources. So what happens in the anode is this organic substrate, such in this case acetate, gets oxidized and generates protein. And during this process, they dump electron directly to the electrode surface. Now that proton now goes into the cathode chamber and has in the presence of a electron acceptor, in this case oxygen, gets reduced and generates water in the process of completing the full circuit, and you are getting electricity. So these MFCs, microbial fuel cells, are basically utilizing bacteria as a catalyst to oxidize substrate and produce electrons. And that electrons is basically the source of the electricity we can get directly from this microbial fuel cell process. So what is the mechanism of electron transfer by these microbes? It's not fully understood, but there are generally three routes that are known how these microbes are transferring electrons. One is the direct electron transfer. So these microbes are directly attached to the electrode, transfer electrons. Second one is, recent finding is they're known to produce these very conductive nanomerials that are produced by specific microbes, and these electrons are making to the electrode using these nanowires, and they are also mediators that are generated by these microbes that can then transfer electrons to the surface of the electrode. There are a few very well-known pure cultures, such as Frontal and Geobacter, which has been studied extensively for these sorts of mechanisms. So how does a typical microbial fuel cell look like? In the lab scale, you see a typical two-chamber MFC system, as well as a single-chamber MFC system. There are a number of miniaturized microbial fuel cells that has been developed for a variety of different applications, such as for powering a remote sensor, or building a high-throughput screen tool, which I will talk a little bit more in detail later on. There has been also quite a bit of effort in moving and trying out this one in a pilot scale plant. So you can see some examples of pilot scale microbial fuel cell for brewery waste treatment. The Office of Naval Research in the United States is interested in producing electricity from the sea. There's also some treatment waste microbial electrolysis cell to generate hydrogen gas out of these systems. So where is currently the efficiency, electricity generation capability of these MFCs. As you can see, there have been dramatic increase in the number of reports as well as the performance increase in recent years. And now it is reaching to the level of where it finally potentially makes economically feasible to use microbial filter as a electricity producing or electricity harvesting system. Now, so within the context of a wastewater management process, where do people think of this MFC will fit? And in most cases, people are considering putting this MFC in a anaerobic digester for energy generation, organic removal, and sludge reduction. The main reason why people are interested in acetate conversion in this MFC is because acetate is a sort of the product that cannot be fermented further by microbes, and that's one of the reasons why tremendous effort has been put in to convert acetate and utilize the substrate. Now, let me briefly also introduce the other the bioelectrochemical system that are of high interest in recent days. So this microbial electrolysis cell is also a bioelectrochemical system where minimal electrical power is provided to achieve hydrogen or chemical production. And the overall architecture is almost identical as the microbial fuel cell. So it still has an anode cathode chain compartment separated by a proton exchange membrane. Anode reaction is same. The only difference here is on the cathode side, in the absence of oxygen, so in the absence of electron acceptor, that proton can be directly converted into hydrogen. Now, there is one problem, or one challenge here. The challenge here is that it is not a spontaneous process. So if you look at the redox potential between anode and cathode in this MC, you still need theoretically you need about 140 millivolt applied to the electrode for the proton to convert into hydrogen. However, if you compare this one to a straight water electrolysis, which in theory requires about 1.2 volt, this is a significantly lower voltage input that you need to have a hydrogen generation. In practice, so what are the numbers in practice? In practice, you do need about 0.25 or 0.2 to 0.25 volt to get hydrogen production in MEC, compared to about 1.8 volt that you need for water electrolysis-based hydrogen production. Now, why are we interested in hydrogen? So hydrogen is interesting in a variety of different scales, anywhere from industrial, large scale to a small scale. First of all, it is a very intensive energy carrier, especially compared to methane. So this is one of the main interests why hydrogen is of great interest in many different applications. There is no emission of greenhouse gas, there's an added benefit. It is also a raw material. Hydrogen itself, other than direct fuel, is also a raw material for anything from power and fuel cell to aerospace application, production and fabrication of varieties of metals, recovery and refining, the production of fertilizer. So hydrogen has many different uses other than just a straight fuel. Now, if you look at the production cost of hydrogen using conventional technology, so non-biomass-based technology, is a range of production costs. It's a little bit difficult to compare this directly with a microbial electrolysis cell-based hydrogen production. So let me compare just the water electrolysis versus microbial electrolysis cell. The United States Department of Energy is targeting a cost about $3 to $4, a few dollars per kilogram of hydrogen is sort of the current target. So as you can see from here, that MEC has a much better, much lower cost compared to water electrolysis, mainly because of the much lower energy input that you need for hydrogen generation. So specifically, why a microbial electrolysis-based hydrogen production? So first of all, we all know that hydrogen can be biologically produced by bacterial fermentation. The maximum hydrogen you can produce from a hexose is 12 mole of hydrogen per 1 mole of hexose. So that's a theoretical maximum that you can get. However, only about 4 mole of hydrogen per mole of hexose can be converted in a regular fermentation process. In practice, it's more like 2 to 3 mole of hydrogen. So how can you recover the residue, the missing hydrogen that cannot be recovered by the bacterial fermentation? And this is why MEC is capable of producing almost close to a theoretical limit. So if you look at the MEC performance for hydrogen production, you can see that some of the reports are reaching close to 11 mole of hydrogen production, and that's very close to a theoretical limit. If you can produce more than about around 10 mole of hydrogen per mole of hexose, then the normal thought is that is the range where it's becoming economically feasible. But one of the issues here is that it is not a self-sustainable process. It's not a zero energy input process where you still require a little of external power for hydrogen production, but however it requires much less energy than water electrolysis. So our overall vision in this area is that we would like to have a hybrid system that couples this microbial fuel cell for electricity production, couple that into a microbial electrolysis cells for hydrogen production. So this hybrid system will take the wastewater as an inflow into a system, and if electricity is the output that we would like to have, utilize the electricity that is directly generated from microbial fuel cell. At the same time, alternatively, you can feed the electricity that is generated from microbial fuel cell, feed it into the microbial electrolysis cell to apply that 0.2 or 0.25 volt that you require for generating hydrogen as a clean burning fuel. So this is our overall vision, and this is as well. This is also the goal of our Gates Foundation GC Round 7 grant. So what is the biggest challenge in getting this one as a self-sustainable simultaneous wastewater treatment and electricity as well as biohydrogen production? You still need a sufficient high power out of the MFC at low cost. And there are a lot of development that needs to be still done in the variety of different aspects of this microbial fuel cell, as well as the microbial electrolysis cell. So because there's similarity in the system, if you can come up with a system that produces high electricity, that technology can almost directly be applied into an MEC because those systems are almost identical except for the cathode power. So there are a lot of challenges in this bioelectrochemical system in general, and all of these challenges, whether it's an anode, cathode, or protein exchange membrane, it all comes down to cost and efficiency. So let's look at the cost factor here of these conventionally used materials that goes into microbial fuel cell as well as the microbial electrolysis cell. Variety of different carbon-based anodes are developed, and this carbon-based anode is what makes NFCs cheap enough to start with. You can see that this cost for the variety of different anode material. You also see a much bigger cost for this ion exchange membrane. However, this problem is at this point solved because you need an ion exchange membrane if you're working with a two-chamber system. However, people are currently using a single-chamber system using an air cathode, which does not require this ion exchange membrane. So this problem here is pretty much a solved problem now. If you look at the cathode, your cost is almost 2 orders of magnitude higher than anode. So at the end, what we need is both a new material development for anode to further reduce the cost and improve the efficiency, as well as working on cathode to lower the cost and while you maintain the highly efficient process. So our strategy in enabling this high power, high electricity generation in this microbial fuel cell is based on maximizing the reactions and electricity generation by using a multi-dimensional macro to nanoscale electrodes. So we would like to incorporate this multi-length structure into our electrode. Start out with this 3D structure. For example, you can use carbon fiber boards, you can use stainless steel mesh and styrofoam. There are a variety of different macroscale 3D structures that you can have. These macrostructures could have this macro scale or micrometer scale, from few to few tens of micrometer scale porous structure. And then within this porous structure, our strategy is to incorporate nanoscale carbon nanotubes bundles or other nanomaterial to have basically a better conduit for electron transfer. And then also on the cathode side have nanoparticle-based catalysts to have a high efficiency. So using this approach, you can get extremely large surfaces for high reactions, as well as this multi-scale dimension for excellent electricity generation and collection would be possible. So this is our overall strategy in enabling this MFC hybrid system. So first, our approach is there are a lot of testing needed at every step of this MFC development. So first of all, we actually develop a high-throughput screening system so that we can test a variety of different conditions, variety of different materials, as well as a variety of different operating conditions. And using that screening system, we develop then this anode cathode material. So you can take a single MFC and you set up many of these, or you can take a single MFC, miniaturize it, and making it as a high-throughput array, just as DNA microarray has changed the scope of how screening is being done. So basically it's a miniaturized microbial fuel cell, 24 of them in a small 2x3-inch glass slide. This is how the device looks like. This is our basically high-throughput screening platform. So we utilize this system to screen for environmental microbes. So initially we collect some samples, do pre-screening of these electrolytes. So what we're trying to do here is find highly efficient electrochemically active microbes from the environment. We're doing a primary screen using our MFCs to look at the power density, and we found some interesting microbes. We then do a confirmation screen using our screening device, as well as validation using a conventional system, doing it in sequencing what particular microbes are actually producing this high power. We currently have a 96+ screen system for microbial fuel cell, direct microbial fuel cell applications. This is how this system looks like. I'll probably skip this slide at the time. Now, using this screen system, we are screening these different materials that we have developed. So first of all, we start out with a 3-dimensional porous structure, such as stainless steel mesh, and our method is directly synthesizing carbon nanotube on this low-cost stainless steel material. So we are obviously not the first one who started making nanomaterial-based anode. However, our approach is very different. If you directly, let me just skip this slide for the time. So the part where our approach is very different from other people is most of the people basically dip their electrode into some carbon-based nanomaterial and rely on the physical attachment. And you see an improvement in power, but not very much. So as you can see from our own experiment here, if you dip this brush structure, you do see about 2 times improvement in power. However, that's certainly not sufficient for highly efficient electron transfer. So we directly synthesize stainless carbon nanotubes in a stainless steel mesh. This is how the carbon nanotubes synthesized in the stainless steel mesh look like. And we are doing initially a 3D versus 2D comparison. So we're comparing a 3D mesh structure versus a carbon plus structure. And you can see that 3D indeed shows higher power than 2D. And now we're comparing the power density of the CNT synthesized porous structure to this 2D structure without the carbon nanotube. And as you can see, the CNT is directly grown on the stainless steel mesh, shows almost more than 3,000 times high power, high electricity generation compared to a bare electrode. And note that this is using a pure culture. We are currently investigating what physical properties of this nanomaterial is resulting in higher power, because again, most of the current studies that has been going on simply uses variety of different material and make an observation-based report that we try these materials and we sell these type of a high power generation. So we would like to do a systematic study in what properties of this nanomaterial really influences the power generation. So this is the electrodes before the MF0 and after the MF0, where you see a biofilm formation. We have also applied this strategy into growing carbon nanotubes on the other three substrates, such as a stainless steel form. As you can see here, before carbon nanotubes, after carbon nanotubes, and this is again a significant improvement in power output compared to a bare electrode. Where are we going from here? Our next step is using wastewater. The reason we have used a pure culture is to do this screening and to do a systematic comparison. We don't want to have a fluctuation that is coming from the microbes, and that's why we initially did older study using the pure culture. We are not too worried about moving into wastewater because there is a long range of literature in MFCs that utilizes wastewater as a microbial fuel cell anode material, and that shows, as you can see, pure culture versus wastewater. Wastewater almost always shows a significantly higher power generation compared to pure culture. So we are relatively confident that our system will show a much higher power output when we move to the wastewater or mixed culture compared to a pure culture that we have been using in this comparison. On the capital side, we have also started to look into non-platinum catalysts because the use of platinum is probably one of the biggest costs in microbial fuel cell. So we have tried different metal-based product and carbon-based product. What you're seeing here is the direct adhesion of these different nanomaterials on these electrodes. And what you're seeing here is, in this case, we have been using it in the system in a MEC for hydrogen production. What you can see here is that these hydrogen evolution using a nickel or copper has a very high efficiency, very close to platinum in terms of the hydrogen evolution. So this is a very promising result for us that there is a potential of using a non-platinum catalyst. Now this is very different from other type of hydrogen fuel cell that requires a very fast reaction rate. In microbial fuel cell, the rate limiting step is the microbe, not on the catalyst side. So you can, what this one shows is, even though you're not using platinum, you can get a very high efficiency at a significantly lower cost. So what is our conclusion here? We have developed a unique microfluidic system approach to allow high-field screening of this variety of different microbes, variety of different anode materials, as well as we are moving into a cathode material development. Carbon nanotubes directly grown on stainless steel mesh electrode shows significantly higher electricity. The modest cost increase for the synthesis process. People think, oh, a carbon nanotube, that's going to be a very expensive process, but it is actually not. Look at the current lab scale cost of making this one. This will be less than $10 per meter square. Obviously, if you scale up, this cost will probably come down. You're really not adding that much of a cost into the synthesis process. And the non-platinum catalyst has been also shown promise in both MSC for electricity generation as well as MEC for hydrogen production. Where's our future goal in the next 6 to 9 months? I would say right now we are in the technology level of 3 to 4, so we are still in a fairly early stage of technology development. We would like to do a full analysis in optimizing this carbon nanotube synthesis process, apply the system using wastewater, which is actually ongoing right now in our lab. Apply this same system for a MEC-based hydrogen generation, and the only difference, as I mentioned, is you just need to remove all the oxygen making in a anaerobic condition. That's what we need to do to get a hydrogen production, and then applying this non-platinum catalyst and further optimizing it, and to bring both the cost down as well as testing the wastewater situation. So where do we think that this fits in terms of the in the country? Currently, there is biogas, as we all have seen, is currently using as a For example, in the cooking field is an example of the efforts ongoing in Nepal. So this is, for example, where we think our hydrogen gas could make it, or if hydrogen gas is not desired, we can maybe directly use the electricity out of the microbial fuel cell. So from a lab scale emissions to a pilot scale MEC for hydrogen production, I think our approach is combining these microbial fuels and microbial electrolysis cell to get both electricity and hydrogen output would be a potential sort of midterm solution in this wastewater management. So with that, I would like to thank the postdoc and students who worked on this project. This particular work was funded by the Melinda Gates Foundation as well as the United States National Science Foundation. And I would like to also acknowledge some of our collaborators here. Thank you.